For millennia, human evolution has been a slow, natural process, driven by random mutation and environmental pressure. But a new era is upon us, one where humanity is gaining the power to direct its own biological destiny. Scientists are no longer merely observing life, they are beginning to write its code. This unprecedented capability, fueled by advancements in gene editing and synthetic biology, promises to redefine what it means to be human, offering a future free from disease, even extending our lifespan, and adapting us for entirely new worlds. But with such power comes profound ethical questions. At the heart of this revolution lies CRISPR-Cas9, a molecular tool first adapted for gene editing by researchers like Jennifer Doudna and Emmanuelle Charpentier. Described as molecular scissors, CRISPR allows scientists to precisely cut and paste DNA sequences with unprecedented accuracy and ease. This technology, derived from a bacterial defense system, enables the targeted modification of genes, opening pathways to correct genetic errors that were once considered irreversible. Its simplicity and effectiveness have rapidly transformed genetic research and therapeutic development. Beyond simply editing existing genes, synthetic biology takes a more audacious step. It aims to design and construct entirely new biological parts, devices, and systems, or to redesign existing natural biological systems for useful purposes. As researchers at institutions like MIT and Stanford explain, it's akin to programming a computer, but with living cells as the hardware. This discipline combines principles from engineering, biology, and computer science to create novel biological functions, from new forms of medicine to custom-designed organisms. It's about building life from the ground up. For millions worldwide, life is profoundly shaped by genetic conditions, inherited disorders that can cause debilitating symptoms, chronic illness, and shortened lifespans. Diseases like cystic fibrosis, Huntington's disease, and sickle cell anemia are written into an individual's DNA, often with no effective cure. Medical science has long sought ways to alleviate symptoms, but the root cause, the faulty genetic code, remained largely untouchable. This reality has driven decades of research into genetics, seeking not just treatments, but true, fundamental corrections. CRISPR's precision offers a powerful new weapon against these single-gene disorders. Researchers are exploring its potential to directly correct the specific mutations responsible for conditions like sickle cell disease, which affects millions globally. By replacing the faulty gene segment with a healthy one, the aim is not just to manage symptoms, but to provide a permanent cure. Early clinical trials are underway for several conditions, showing promising results in correcting genetic defects in patients' own cells, particularly in bone marrow for blood disorders. Another devastating target is Huntington's disease, a progressive neurodegenerative disorder caused by a single expanded gene. While not curable today, scientists are using CRISPR to silence the faulty gene, preventing it from producing toxic proteins that destroy brain cells. This approach, as discussed by neurologists, moves beyond symptom management to address the disease at its genetic origin. Early-stage research in animal models has demonstrated the feasibility of reducing the harmful protein, sparking hope for human therapeutic applications in the coming years. Inherited forms of blindness, such as Leber congenital amaurosis (LCA), are also on the cusp of being addressed through gene editing. Several studies have shown that by delivering CRISPR components directly to the eye, researchers can correct mutations in genes vital for vision. Ophthalmologists note that the eye is an ideal target for gene therapy due to its accessibility and immune-privileged status, meaning the body is less likely to reject the new genetic material. These treatments offer the potential to restore sight, transforming lives affected by these specific genetic conditions. Synthetic biology extends beyond merely repairing genetic flaws. It ventures into programming cells to perform entirely new functions. Researchers at Harvard Medical School are designing smart cells that can detect early signs of cancer or infection and then deploy therapeutic agents, essentially turning cells into microscopic doctors. This approach represents a paradigm shift from traditional drug delivery, as the cells themselves become dynamic, responsive biological machines, continuously monitoring and intervening within the body, offering a personalized and highly targeted form of medicine. In the fight against cancer, synthetic biology and CRISPR are creating new frontiers. Scientists are engineering immune cells, specifically T cells, to more effectively identify and destroy cancer cells. This process, known as CRT cell therapy, involves taking a patient's T cells, genetically modifying them to recognize specific cancer markers, and then reinfusing them. CRISPR streamlines this process, allowing for more precise and potent modifications, essentially arming the body's own defenses with a highly targeted weapon against malignant growths. Beyond disease eradication, perhaps the most audacious goal of biological engineering is the reversal of aging. For centuries, humans have dreamed of extending life, if not achieving immortality. Modern science, particularly with insights from genetics, now views aging not as an inevitable decline, but as a biological process that might be slowed, halted, or even reversed. Researchers are exploring fundamental mechanisms of aging, from cellular senescence to telomere shortening, believing that genetic interventions could unlock the secrets to a longer, healthier human existence. One key area of focus is telomeres, the protective caps at the ends of our chromosomes. Each time a cell divides, telomeres shorten, acting as a biological clock that limits cell division. Once telomeres become too short, cells stop dividing or die, contributing to aging. Scientists are investigating whether genetically engineering cells to maintain or even lengthen telomeres could extend cellular lifespans. As some geneticists propose, if we can control telomere length, we might significantly delay age-related cellular damage and organ degeneration. Another target in the fight against aging involves senescent cells, zombie cells that stop dividing but remain in the body, releasing inflammatory compounds that damage surrounding tissues and contribute to age-related diseases. Synthetic biology approaches are being developed to program cells to identify and clear these harmful senescent cells. Early studies in mice have shown that removing senescent cells can extend healthy lifespan and alleviate multiple age-related conditions, suggesting a powerful avenue for human intervention. Beyond telomeres and senescent cells, researchers are uncovering entire genetic pathways that influence longevity. Studies of centenarians, for example, reveal specific gene variants associated with extended lifespans and resistance to age-related diseases. Synthetic biologists are now looking to understand and potentially mimic these natural genetic advantages. Could we program our own cells to activate these longevity genes, enhancing cellular repair mechanisms and stress resistance? The potential implications for human health are staggering. As humanity casts its gaze towards other planets, particularly Mars, the challenge of adapting our biology for extreme environments becomes paramount. The Martian surface presents a hostile cocktail of intense radiation, microgravity, and an unbreathable atmosphere. Traditional spacesuits offer protection, but for long-term colonization, scientists at institutions like NASA are considering how to genetically fortify human biology itself. This isn't just about survival, it's about thriving, pushing the boundaries of human adaptability through biological engineering. One of the most pressing dangers on Mars is radiation. Without Earth's protective magnetosphere and thick atmosphere, astronauts are exposed to harmful cosmic rays and solar flares, increasing cancer risk and causing cellular damage. Genetic engineers are exploring how to enhance human cells' natural repair mechanisms or introduce genes from radiation-resistant organisms, like tardigrades, to make humans more resilient. The goal, as posited by some astrobiologists, is to reduce reliance on heavy shielding, enabling lighter spacecraft and habitats, and ultimately, safer long-duration missions. Life in microgravity leads to bone density loss, muscle atrophy, and cardiovascular issues. While countermeasures like exercise are critical, they are not always sufficient. Synthetic biologists envision engineering human physiology to better withstand these effects. This could involve modifying genes related to bone formation, muscle protein synthesis, or red blood cell production to counteract the deleterious effects of a low-gravity environment. As researchers in space medicine suggest, the aim is to minimize the biological cost of space travel, making it more sustainable for future generations. Long-duration space missions also require extreme metabolic efficiency. Every ounce of food and water is precious. Synthetic biology could engineer human metabolism to utilize nutrients more efficiently, perhaps even enabling certain cells to synthesize essential compounds currently obtained from diet. This could reduce logistic burdens and increase self-sufficiency for deep space colonization. Experts in space agriculture are also exploring engineered microbes to supplement human nutrition, a fascinating synergy between biological engineering and humans and their environment. The sum of these capabilities, eradicating disease, reversing aging, and adapting for new worlds, points towards a future where the very definition of human may expand. Transhumanist thinkers and some futurists envision a post-human stage of evolution, where our species actively directs its genetic makeup to achieve capabilities far beyond our natural biological limits. This isn't science fiction to them, it's a logical progression of scientific advancement. The tools are emerging, prompting a profound societal discussion on where the boundaries should lie. This is where the excitement of scientific progress meets the gravest ethical concerns. The ability to erase genetic diseases like cystic fibrosis is widely celebrated. But what about enhancing cognitive abilities, physical prowess, or even aesthetic traits? Where is the line between therapy and enhancement? Ethicists argue this distinction is crucial. Curing illness restores health, enhancement seeks to create better humans, potentially leading to unforeseen consequences, societal divides, and a fundamental shift in human nature that may not be universally desired. The concept of designer babies, selecting or editing embryos for desirable traits, is perhaps the most emotionally charged aspect of this debate. While screening embryos for severe genetic diseases is already practiced in some clinics, active gene editing takes this a step further. As bioethicists point out, if we edit out predispositions to disease, what prevents us from editing in intelligence, athletic ability, or even eye color? This raises concerns about eugenics, genetic discrimination, and a potential future where only the wealthy can afford to give their children a genetic advantage. These ethical boundaries were starkly tested in 2018 when Chinese scientist He Jiankui announced he had created the world's first gene-edited babies. He used CRISPR to modify the CCR5 gene in human embryos, aiming to confer HIV resistance. This rogue experiment, widely condemned by the global scientific community, underscored the urgent need for international consensus and regulation. It ignited a fierce debate about germline editing, changes that would be heritable by future generations, and the severe risks of proceeding without robust ethical oversight and public discussion. The He Jiankui case highlighted a critical distinction: somatic cell editing versus germline editing. Somatic editing targets cells that are not passed on to offspring, like blood cells to treat sickle cell anemia. Germline editing, however, modifies embryos, sperm, or egg cells, meaning the changes are heritable and would affect all future generations. While somatic editing for therapeutic purposes is proceeding, most countries have placed moratoriums or outright bans on germline editing due to the profound, irreversible, and unpredictable consequences it entails for the human gene pool. Even with the precision of CRISPR, there are risks. Off-target edits, unintended changes to other parts of the genome, can occur, potentially leading to new diseases or disrupting vital biological functions. The long-term effects of germline edits are entirely unknown, raising concerns about unpredictable changes to the human species. Biologists emphasize that the genome is an incredibly complex system, and even small, seemingly beneficial changes could have cascading effects throughout generations, altering human health or evolution in ways we cannot currently predict or reverse. Beyond scientific risks, ethical concerns also revolve around equity and access. If gene editing therapies and enhancements become available, who will benefit? The cost of such advanced medical interventions is likely to be extremely high, at least initially. This raises the specter of a genetically privileged class, exacerbating existing social inequalities and creating new forms of discrimination based on genetic makeup. As public health advocates argue, if gene editing can cure disease, it should be accessible to all, not just an elite few. A common ethical argument against human enhancement is the slippery slope. Proponents of this view warn that even seemingly benign genetic interventions for medical purposes could pave the way for increasingly complex and ethically dubious enhancements. Where does the drive to eliminate disease end, and the desire to create perfect humans begin? Critics of this argument say it's a fallacy that presumes a lack of moral agency and regulatory control, but it nonetheless highlights the need for constant vigilance and clear ethical frameworks as the technology progresses. Recognizing the gravity of these issues, international bodies and scientific academies are working to establish global guidelines and foster public dialogue. Organizations like the World Health Organization and National Academies of Science have convened experts to debate the ethical, legal, and social implications of human gene editing. Their goal is to develop a framework that balances scientific progress with responsible innovation, ensuring that these powerful technologies serve the good of all humanity, rather than leading to harmful or discriminatory outcomes. The challenge for regulators is immense. Gene editing and synthetic biology are advancing at an astonishing pace, often outstripping the ability of legal and ethical frameworks to keep up. Different countries have varying laws and moral perspectives, making international harmonization difficult. Legal scholars emphasize the need for adaptive regulatory models that can evolve with the science, rather than rigid rules that quickly become obsolete. This requires ongoing dialogue between scientists, ethicists, policymakers, and the public. Informed public engagement is crucial for navigating this complex landscape. The decisions surrounding human gene editing are not solely for scientists or ethicists, they affect everyone. Educating the public about the potential benefits and risks, fostering open discussion, and allowing diverse voices to be heard are essential steps in building societal consensus. Without broad understanding and participation, there is a risk of either fear-driven prohibitions that stifle beneficial research or unchecked advancement that disregards ethical boundaries. While CRISPR has dominated headlines, the field of gene editing continues to evolve rapidly. Newer tools, such as prime editing and base editing, offer even greater precision, allowing for single base changes or small insertions/deletions without cutting the DNA double helix. These next-generation technologies promise to address some of CRISPR's limitations, including off-target effects, and expand the range of genetic conditions that can be corrected. Researchers anticipate a suite of highly refined tools, each suited for different genetic interventions, pushing the boundaries of what's possible even further. Synthetic biology also holds the promise of constructing entire genomes from scratch. Pioneers like J. Craig Venter have already created synthetic bacterial genomes, demonstrating the feasibility of designing and building entire genetic operating systems for living organisms. The extension of this technology to more complex cells, and eventually perhaps human cells, is a long-term, highly ambitious goal. This could enable unprecedented control over cellular function, opening avenues for extreme bioadaptation, or even designing completely new forms of life with specific desired characteristics. Beyond editing, synthetic biology is exploring cellular reprogramming, the ability to convert one cell type into another. For instance, skin cells could potentially be reprogrammed into neurons or pancreatic cells to treat neurological disorders or diabetes. This avoids the ethical complexities of embryonic stem cells and offers a personalized source of therapeutic cells. Researchers see this as a way to regenerate damaged tissues and organs within the body, effectively reversing disease and age-related decline at a foundational cellular level, without external genetic modification of the entire organism. The confluence of CRISPR and synthetic biology strongly points towards an era of highly personalized medicine. Genetic therapies could be tailored precisely to an individual's unique genetic makeup, offering treatments that are more effective and have fewer side effects than current broad-spectrum drugs. Imagine a future where your genetic predispositions are analyzed, and custom-designed gene therapies are deployed to prevent diseases before they even manifest, or to treat them with unprecedented accuracy. This represents a fundamental shift in healthcare philosophy, from reactive to proactive and preventive. Some researchers are even cautiously exploring the concept of a therapeutic gene drive for human health, though this remains highly controversial and largely theoretical. A gene drive system ensures that a particular genetic trait is inherited by nearly all offspring. While primarily researched for controlling disease vectors like mosquitoes, hypothetically, if a severe genetic disease could be eradicated from a population by ensuring a corrected gene spreads, it raises enormous ethical questions about altering the human germline on a population scale. The risks of unintended consequences are immense, making this a territory scientists approach with extreme caution. As we discuss engineering the human species, it forces us to confront fundamental questions about what constitutes human nature. Is it merely our genetic code, or something more? If we gain the power to alter our biology, what aspects should remain untouched? Philosophers and theologians debate whether there are essential human qualities that should not be subject to genetic manipulation. This isn't just a scientific or medical discussion, it's a profound cultural and existential one, challenging centuries of self-understanding and raising concerns about the sanctity of life. The advances in CRISPR and synthetic biology are not just changing what's technically possible, but also what we dare to imagine. From a future free of inherited illness to humans living on Mars, the potential applications are vast. This technology forces us to move beyond passive observation of evolution and into active participation. As some leading scientists suggest, we are becoming the architects of our own biology, wielding tools that were once the domain of science fiction. The sheer scale of this transformation requires both courage and profound humility. Amidst the excitement, a significant concern is genetic determinism, the idea that our genes solely dictate who we are. While genes play a crucial role, environmental factors, lifestyle, and individual choices also profoundly shape human development and health. Overemphasizing genetic solutions might lead to neglecting social determinants of health or creating a false sense that all problems can be edited away. Sociologists caution against reducing human identity to a mere sequence of DNA, emphasizing the complex interplay of nature and nurture. The promises of personalized medicine and genetic cures hinge heavily on accessibility and affordability. Current gene therapies can cost millions, putting them out of reach for most of the global population. Researchers are actively working on ways to reduce these costs, from more efficient production methods to new delivery mechanisms. However, policymakers in healthcare systems face the daunting task of integrating these advanced therapies into public health frameworks, ensuring equitable access and preventing the creation of a two-tiered healthcare system where genetic advantages are bought, not earned. The pursuit of the perfect human through genetic engineering is fraught with peril. What constitutes perfection is subjective, culturally bound, and constantly evolving. Attempting to engineer specific traits could inadvertently reduce human diversity, a cornerstone of our species' resilience and adaptability. Moreover, what seems desirable today might have unforeseen disadvantages in a future environment. Biologists often highlight that genetic diversity is a strength, providing the raw material for adaptation to new challenges, a principle that could be undermined by overly narrow selection. Around the world, legal frameworks are slowly beginning to catch up to the science. Countries like the United Kingdom have robust regulatory bodies, while others are still grappling with how to classify and control gene editing technologies. The European Union has taken a more cautious approach, often classifying CRISPR-edited organisms similarly to genetically modified organisms. In contrast, the United States has adopted a more product-by-product regulatory stance. This patchwork of regulations highlights the need for international cooperation to prevent ethics shopping, scientists moving to jurisdictions with laxer rules. Biotechnology companies are at the forefront of translating these scientific breakthroughs into commercial applications. Billions of dollars are being invested in developing CRISPR-based therapies and synthetic biology platforms. These companies face the dual challenge of pioneering new treatments while navigating complex regulatory landscapes and ethical considerations. Their pursuit of profit often intersects with the public good, necessitating transparent research practices and a commitment to responsible innovation, ensuring that these powerful tools are developed with societal well-being in mind. Alongside commercial ventures, academic research remains a vital engine of discovery. Universities and public institutions often drive the foundational science, operating within frameworks that prioritize open science and peer review. Researchers emphasize the importance of sharing methodologies and findings to accelerate progress and ensure scrutiny. This collaborative, transparent approach contrasts with some commercial practices, and its continuation is critical for fostering a robust understanding of these technologies, allowing the entire scientific community to collectively address challenges and validate results. A continuous area of research and concern is the diligent monitoring and mitigation of off-target effects. Even with advanced CRISPR systems, unintended edits can still occur, potentially leading to unforeseen health consequences. Scientists are developing increasingly sophisticated computational algorithms and experimental techniques to predict and detect these off-target edits with higher accuracy. The goal is to refine gene editing to a level where its precision is absolute, minimizing any risk of collateral genetic damage. This ongoing refinement is essential for ensuring the safety and long-term viability of human gene therapies. A critical practical challenge in gene therapy is the efficient and safe delivery of gene editing tools into target cells within the human body. Viruses, particularly adeno-associated viruses, AAVs, are currently the most common vectors, engineered to deliver the genetic payload without causing illness. However, concerns remain about immune responses and potential limitations in targeting specific tissues. Synthetic biologists are developing non-viral delivery methods, such as lipid nanoparticles or engineered exosomes, to overcome these challenges, aiming for safer, more targeted, and repeatable delivery of gene editing components. As synthetic biology progresses, so too do concerns about biorisk and biosecurity. The ability to engineer pathogens or create new biological threats is a serious consideration. Governments and scientific communities are working to establish robust biosecurity protocols, including strict oversight of gene synthesis companies and research facilities. The dual-use nature of these technologies, their potential for both immense good and catastrophic harm, necessitates vigilant monitoring, ethical guidelines for researchers, and international agreements to prevent misuse, particularly by malicious actors or state-sponsored programs. Beyond the scientific and ethical debates, humanity faces the profound challenge of societal adaptation. How will our societies change if aging is significantly slowed, or if humans can be engineered for specific tasks or environments? Social scientists predict shifts in demographics, economic structures, and even interpersonal relationships. These technologies force us to re-evaluate deeply held values in institutions, from healthcare systems to education, and consider the broader implications for human civilization. It's a journey into uncharted territory, demanding foresight and collective wisdom. The engineering of the human species is not just a scientific problem, it's a philosophical one. What is the meaning of life, of suffering, of death, if these biological realities can be modified? Does the elimination of genetic flaws diminish what it means to be human, or enhance it? These questions, once confined to abstract thought, are now becoming tangible. Ethicists, philosophers, and spiritual leaders are increasingly engaging with scientists to explore these profound dimensions, seeking a moral compass in an era of unprecedented biological control. The implications for human reproduction are particularly acute. In vitro fertilization, IVF, already allows for selection of embryos, but gene editing could enable proactive modification before implantation. What decisions will prospective parents face? The ability to potentially eradicate diseases for their children and future generations is a powerful motivator. However, the line between preventing disease and choosing desirable traits will be blurry, raising new questions about parental responsibility, reproductive autonomy, and the rights of future, unedited individuals. As gene editing becomes more accessible and precise, discussions will inevitably shift beyond medical necessity. Could gene editing be used for non-medical enhancements, such as improving memory, increasing strength, or altering personality traits? While currently theoretical and ethically contentious, the underlying technologies continue to develop. The question for society becomes whether these applications are merely a logical extension of our desire for improvement, or a dangerous step towards commodifying human biology and exacerbating existing inequalities, creating a form of genetic capitalism. The aspiration to engineer humans for extreme environments like Mars underscores a fundamental shift in our relationship with nature. Instead of adapting our environment to suit us, we are now contemplating adapting ourselves to suit the environment. This represents a bold, almost audacious, human endeavor. It speaks to our species' inherent drive to explore and conquer new frontiers, but also raises questions about ecological responsibility and whether we should fundamentally alter ourselves rather than finding ways to protect and cherish the terrestrial environment that shaped us. The speed of scientific discovery often outpaces ethical preparedness. We find ourselves with immensely powerful tools before fully understanding their long-term implications or establishing a collective moral framework for their use. This requires proactive, rather than reactive, engagement from all sectors of society. Bioethicists argue that we must build robust ethical scaffolding concurrently with scientific advancement, ensuring that our technological capabilities are always guided by a deep sense of responsibility and foresight, rather than being driven solely by curiosity or commercial interest. As we stand on the precipice of engineering the human species, it's vital to consider our long-term vision for humanity. What kind of future do we want to build? One where genetic diseases are a distant memory, where human lifespan is extended, and where we can journey to the stars? Or one riddled with genetic inequality and unforeseen biological consequences? The answers will shape not just individual lives, but the very trajectory of our species for millennia to come. It requires a thoughtful, inclusive, and globally coordinated approach. Unlike other technologies, genetic engineering leaves an indelible mark, potentially altering the very essence of future generations. This permanence adds layers of responsibility unmatched by previous scientific advancements. Scientists now hold in their hands the blueprint of life itself, with the capacity to modify it in ways that echo through time. This profound power demands not just technical mastery, but also deep ethical reflection, humility, and a commitment to safeguarding the inherent dignity and diversity of human life, even as we seek to improve it. The journey into engineering the human species is still in its early stages. Breakthroughs continue to emerge at an exhilarating pace, offering both tantalizing promises and formidable challenges. From curing intractable diseases to redefining human longevity and adaptability, the possibilities seem boundless. Yet, the path ahead is not clear-cut. It requires continuous scientific exploration, rigorous ethical debate, broad societal engagement, and thoughtful regulation. Humanity stands at a crossroads, where the power to reshape itself is no longer theoretical, but increasingly real. In the immediate term, CRISPR is making its most significant impact in treating devastating genetic diseases. Clinical trials for conditions like beta thalassemia and sickle cell disease are showing very encouraging results, moving from experimental research to potential therapies. Researchers are also exploring its use in treating certain cancers by engineering immune cells, as we discussed. These applications, widely considered therapeutic, represent the low-hanging fruit where the ethical consensus is strongest, using gene editing to alleviate suffering and restore health, a goal that few would argue against. Synthetic biology also holds immense therapeutic promise. Beyond cancer, it's developing engineered probiotics for gut health, smart cells for targeted drug delivery in chronic conditions, and even biosensors that can detect disease onset much earlier. These applications focus on optimizing the body's own systems or introducing novel biological functions for health benefits. As researchers from leading institutions suggest, synthetic biology could allow us to build new biological tools that go beyond what natural evolution has provided, creating personalized, dynamic, and highly effective medical interventions. The convergence of CRISPR and synthetic biology has significantly accelerated aging research. Instead of simply studying the effects of aging, scientists can now actively intervene in the biological processes that cause it. Genetic interventions targeting senescent cells, telomere maintenance, and metabolic pathways are moving from theory to experimental validation in animal models. While human trials for age reversal are still nascent and highly scrutinized, the fundamental understanding and the tools to manipulate these processes are developing at an unprecedented rate, offering a potential future where healthy longevity is the norm. Even the reversal of aging markers raises ethical questions. If lifespan is significantly extended, what are the societal implications for resource distribution, population growth, and retirement systems? Who would have access to these life-extending therapies? ethicists warn that a prolonged lifespan for a select few could exacerbate existing inequalities. There's also the philosophical question of whether endless life is truly desirable, and what constitutes a meaningful human existence. These discussions must run parallel to scientific progress, shaping how and for whom these life-altering technologies are deployed. Engineering humans for Mars is a distant goal, but incremental steps are already being explored. NASA funds research into enhancing astronaut resilience through genetic means for specific challenges like radiation. Early applications might involve gene therapies to boost antioxidant defenses or improve DNA repair. These are seen as protective measures, rather than radical enhancements, aiming to mitigate known risks of space travel. The transition from therapy to enhancement, however, remains a critical threshold, demanding clear guidelines and international collaboration to ensure responsible development. The global nature of gene editing and synthetic biology necessitates global governance. As the He Jiankui case demonstrated, actions in one country can have profound international repercussions. Scientists, policymakers, and ethicists from around the world are calling for shared ethical principles, common regulatory standards, and mechanisms for international oversight. Without such frameworks, the risk of a fragmented, unregulated landscape increases, potentially leading to dangerous experimentation or the proliferation of unethical practices that could affect all of humanity. Ultimately, the direction of human genetic engineering will be shaped not just by scientific capability, but by public discourse. It's imperative that these complex topics are discussed openly, thoughtfully, and inclusively across all segments of society. Public forums, educational initiatives, and media engagement play a crucial role in demystifying the science and enabling informed civic participation. The future of our species, and the ethical boundaries we choose to draw, depend on our collective ability to understand, debate, and ultimately decide how we will wield this extraordinary power. A critical distinction in the ethical debate centers on interventions at the individual level versus those that could impact the entire species. Curing an individual's genetic disease is one thing, making heritable changes to the human germline that could alter the entire gene pool is another. The latter carries a far greater burden of responsibility and potential risk. As various expert panels have concluded, such species-level interventions require a level of societal consensus and understanding of long-term consequences that we are far from achieving. Looking ahead, the next few decades will be pivotal. We can anticipate significant advancements in gene therapy for genetic diseases, with more conditions becoming treatable. Research into aging reversal will intensify, with early human trials exploring specific markers. The dream of engineering humans for space will likely remain in the realm of research, focused on fundamental biological resilience. The ethical debates will only grow more complex, demanding continuous dialogue and evolving frameworks. The revolution in genetic engineering is here, and its impact will only deepen. The power to engineer the human species is arguably the most profound technological capability humanity has ever developed. It holds the potential to alleviate immense suffering and unlock new frontiers for our species. Yet, it also carries the risk of fundamentally altering human nature in ways we might regret. The ultimate challenge lies in harnessing this immense power responsibly, guided by foresight, empathy, and a deep respect for human dignity and diversity. Our choices today will determine the future of humanity itself. The journey from treating debilitating diseases to enhancing human capabilities is a continuum, not a sharp divide. Where societies draw the lines will be a matter of ongoing debate and evolving consensus. For now, the focus remains primarily on therapeutic applications, offering hope to millions suffering from genetic conditions. But the underlying science for enhancement is often the same, making the ethical distinction crucial. As the tools become more potent, so too does the need for rigorous scrutiny and a global commitment to responsible scientific practice. The ambitious challenge of living on Mars serves as a powerful catalyst for pushing the boundaries of human engineering. The unforgiving Martian environment compels us to think deeply about human biological limitations and how they might be overcome. While terraforming a planet is a long-term dream, adapting human biology itself is a more direct, albeit profound, pathway. It forces a conversation about human resilience, adaptability, and the ethical implications of intentionally altering our species for survival and expansion beyond Earth. The red planet becomes more than a destination, it's a crucible for our biological future. Ultimately, the story of CRISPR and synthetic biology is a story about choice. Humanity now possesses the power to choose, to some extent, its own genetic future. Will we use this power to alleviate suffering, extend healthy lives, and explore new worlds, or will we succumb to the allure of designer traits and exacerbate existing inequalities? The answer is not predetermined. It will be shaped by the decisions we make collectively, the ethical lines we draw, and the values we choose to uphold. The age of engineering the human species has begun, and the dialogue must continue. The ability to edit our genes also impacts our understanding of identity. If elements of our biological selves can be altered, does it change who we fundamentally are? This question resonates deeply with individuals who define themselves by their genetic conditions or unique biological traits. Ethicists emphasize the importance of recognizing the complex interplay between genetics, personal identity, and lived experience, ensuring that efforts to improve do not inadvertently devalue individuals or groups based on their natural genetic makeup. Respect for inherent human diversity remains paramount. The rapid pace of innovation in gene editing technology is both a blessing and a curse. It brings cures faster but also leaves less time for thorough ethical and societal deliberation. Scientists are keenly aware of this paradox, often urging caution even as they push the boundaries of discovery. This tension between accelerating progress and ensuring responsible deployment will define the field for decades to come. It requires constant self-reflection within the scientific community and robust engagement with the public to ensure that innovation serves humanity's best interests. The call for a global bioethics summit, regularly convened, grows louder with each new breakthrough. A dedicated international body, capable of fostering consensus, setting norms, and issuing recommendations, could provide much-needed guidance. Such a summit would involve not just scientists, but ethicists, lawyers, policymakers, religious leaders, and public representatives from diverse cultural backgrounds. This collaborative approach is vital to navigate the complex moral landscape and establish universal principles that can guide the responsible development and application of human gene editing technologies worldwide. As we conclude our investigation, the prevailing sentiment is one of immense hope tempered by profound caution. Hope for a future where debilitating genetic diseases are relegated to history, where extended health spans are common, and where humanity can truly become a multiplanetary species. But caution against the hubris of altering our fundamental biology without fully understanding the long-term consequences, against creating new forms of inequality, and against losing sight of what truly makes us human. The tools are here, but the wisdom to wield them responsibly is still being forged.